Hello students. Today in this video we are going to learn about the cell, the fundamental unit of life, the basic unit of life. In this video we will be learning about prokaryotic and eukaryotic cell, multicellular organism, cell membrane and cell wall, cell organelles, chloroplast, mitochondria, vacuoles, endoplasmic reticulum, Golgi apparatus, nucleus. Chloroplast, mitochondria, vacuole, endoplasmic reticulum, Golgi apparatus, nucleus are the cell organelles which are present inside the cell. And also nucleus, its basic structure and the number of chromosomes. Let's begin with the discovery of cell. Who discovered the cell for the first time? The cell was discovered by Robert Hooke in 1665. He is an English scientist. He saw the thin slice of cork under the microscope, crude microscope. Why it is known as a crude microscope? Because he was not having the developed advanced microscope that we have right now today. In the image that is in the picture that is in your left hand side is the microscope that Robert Hooke has used. He observed a honeycomb-like structure. The image is given on your right hand side, honeycomb-like structure, and named them as cellula or cells. His discovery indicated for the first time that living organism consisted of number of small structures or units, right? So he was a pioneer to discover that all the living organisms are made up of cells. Let's know more about the history of cell. Though the cell was discovered by Robert Hooke, but Anton von Leeuwenhoek was the first one to study living cell. Then came the Robert Brown. He discovered and named nucleus in the cell. Then came the Skevan and Skeleton. They formulated the cell theory. What is a cell theory? Cell theory indicates that it suggests that all the living organisms are made up of cells, and cell is a fundamental basic unit of life. Next, this theory was a bit modified, and the new modern theory is there by Rudolf Virchow. Rudolf Virchow added one statement that omnis cellula e celluli. What does that indicate? What does that mean? Omnis cellula e celluli stands for all the living organism arises from pre-existing cell. So, adding this third point into the cell theory, now we accept the modern cell theory. Modern cell theory has 3 points. Number 1 is all the organisms are made up of cells. Cell is a basic fundamental unit of life. And third point is all the living organisms are made up of cells. Moving ahead. What are living organisms made up of? Just as a building is made up of bricks, the body of all living organisms is made up of cells. Now, organisms on the basis of number of cells present, their organization of the body, either they are categorized as multicellular or unicellular. The examples of unicellular are Chlamydomonas, amoeba, paramecia. And the examples of multicellular are plants, animals, humans, etc. Irrespective, if we talk about unicellular or multicellular, all the cells are performing similar basic function for the survival of an individual, for the survival of an organism. Now, what are the gadgets that we use to study cells? The gadget that we use to study cell is microscope. We can't see the cell with our naked eye, so we study cell with the help of microscope. In the figures, I have given 2 figures. One figure shows the compound light microscope. The other one that is on the right hand side is an electron microscope. The compound microscope, the light microscope that is on the left hand side is used in laboratories. The magnification ranges is 100 to 1500. So the power that the compound microscope magnifies the image around 100 times or 1500 times. Whereas if we wanted to study in depth the cell organelles or more microscopic or smaller microscopic organisms, then we use electron microscope, because its magnification range is from 1 lakh to 5 lakh. So there are 2 types of microscope, light microscope and electron microscope. Moving ahead, One question comes, does all the cells have same shape and size? No. All the cells, there are different types of cells with different shape and different size. Why? It's depending on their specific function. Size and the shape of the cells vary, but all of these are ultimately determined by the specific function. According to their function, they have different shape, they have different size. Some cells can change their shapes like amoeba and WBC, but plants and animals have almost fixed shape. We can't change our shape. The smallest cell is PPLO, that is mycoplasma. The full form of PPLO is pleuropneumonia-like organism. The largest cell on planet is ostrich egg. The longest animal cell is nerve cell. The largest animal cell is egg cell. And a micrometer, it's a unit written. A micrometer or micron is 1,000 of the millimeter. Here is given a bit of, you can say, all right. Now, difference between unicellular versus multicellular. Unicellular, made of single cell able to perform all the life processes like obtaining food, respiration, excretion, growth, and reproduction. Cannot be divided into cells. They are also called acellular organisms. Examples are amoeba and paramecium. On the other hand, multicellular are made up of many number of cells. They have specific, cells have a similar basic structure and similar basic life activities. They have different cells to perform different functions like respiration, excretion, growth, reproduction. Different shapes and aggregated to form different tissues. Tissues form the organs, and organs give rise to organ systems that perform special functions, right? And examples are plant, animal, and fungi. Cell is a basic unit of life. It performs all life. Why it is known as basic unit of life? Because it performs all life functions like intake of food, materials, excretion, metabolism, respiration, irritability, etc. All the functions that we are performing, excretion, metabolism, respiration, digestion, that takes place inside our cell. That's why it's a basic function. Division of labor. What is the meaning of division of labor? Division of labor is cells usually possess a number of components called cell organelles, right? So it's like division of the work. Cell has an organelle. Each and every organelle has a specific role, as a specific function to perform. That is division of labor. Each cell organelles perform different functions. There are a few examples given like clearing of waste material, synthesis of protein, synthesis of lipid, and packaging. There are a few more examples like packaging, production of energy, etc. What is cell made up of? A cell is a tiny mass of protoplasm which is surrounded by membrane and is capable of performing all functions of life. What is this protoplasm? Here in the diagrams, you can see a cell is having cell membrane. It has the cytoplasm and the nucleus. Nucleus plus cytoplasm is termed as protoplasm. Am I clear? Very good. So, what is a cell? A typical cell is formed of three parts. A plasma membrane to create a separate environment. It may be cell membrane, it may be cell wall in some of the organisms. Nucleus for its control. Cytoplasm possessing metabolic machineries. Like all the cell organelles are present in cytoplasm. Moving ahead with plasma membrane, that is a cell membrane. The outermost delicate elastic membranous covering of the cell that separates its contents from the external environment is called plasma membrane. Plasma membrane is selectively permeable. Now, what is the meaning of selectively permeable? It will allow entry of certain substances only. There are the membranes that are known as semi-permeable, there are known as permeable, right? But our plasma membrane is selectively permeable. It will only allow the entry of certain substances only. It is made up of bilipid layer. What is the meaning of bilipid layer? The lipid layer, two lipid layer. Bi always indicates a two lipid layer, and proteins are integrated in out and inside. Proteins are integrated either inside or outside. Small carbohydrates are attached at places to outer surface of lipids and protein. What is its function? Shape of contents of the cell. It gives a shape. Mechanical barrier. It is semi-permeable. It performs endocytosis. It has the recognition centers. It allows the flow of information in the sense flow of things that are they are synthesized inside the cell or that is coming from outer environment. It performs osmosis, cell continuity. It balances and maintains the cell continuity. Modified to perform special function like absorption in microvilli. What are the microvilli? Microvilli are present in the small intestine of humans that allow the absorption of food. This is the structure of plasma membrane. You can see here lipids, oligosaccharides are the carbohydrates, right? These are the lipids, right? Extracellular matrix, and then these are the proteins that are either peripheral or intrinsic proteins, right? This is SEM, plasma membrane, right? That is electroscopic you can say image of plasma membrane. Now, how does the transport across the membrane take place? Through the plasma membrane. Plasma membrane is the basic thing from which the substance from outside the environment enters into the cell or from cell is transported to other cell, right? From one cell to other cell. Membrane acts as a physical barrier between the organelles of a cell and cytoplasm and its surrounding environment, right? Membrane could be impermeable. Impermeable, it will not allow anything to pass from it. Permeable, it will allow everything to pass from it. Semi-permeable, permeable to solvent, but prevents solutes passage. Selectively permeable, if membrane allow the passage of solvent and some selected solute. There are two types of transportation that are taking place inside the cell. One is active transport and another one is passive transport, right? Active transport is when the substance passes by consuming ATP, that is energy. Passive is the transportation without consumption of ATP. Bulk transport, there are different types now. Bulk transport as well. If large amount of substances and food particles across the plasma membrane are transported, such is known as bulk transport. The process of bringing the bulk of material from outside to the formation of endosmosis is known as endocytosis. This word you have to remember, my dear students. That is endocytosis. And is inside, cyto is the cytoplasm, inside the cytoplasm, right? The process when something is taken out from outside external environment inside the cell, a bulk quantity, that is known as endocytosis. Bulk explosion of material as in secretion excretion is called exocytosis. There are the two terms. My dear students, always remember in biology we usually use two words, endo and exo. Endo is always inside, exo is outside, right? X is to leave, fine. So exocytosis, something when bulk is secreted outside, when something is removed from the cell outside, that is known as exocytosis. And when something is entered, right, something is absorbed from external environment inside the cell is known as endocytosis. Now let's see in detail about the passive transport. Passive transport is of two types, diffusion or osmosis. Now here from the figure, I won't be reading it much. Here from the figure, it will be clear to you. There's a membrane, right? Here, the yellow dots are less and on this side, yellow dots are more. What does that indicate? The yellow dots are nothing but these are the solvent molecules, right? So these are the solvent molecules that they move from higher concentration to lower. Always remember the movement of molecule is from higher concentration to lower concentration. From the region of higher concentration to the region of lower concentration. This is known as diffusion. Here one more example is given. Even in fact, you can also do this practical experiment at your home. Just take your ink or your color, take one glass of water or transparent glass bowl and put one drop of ink into it. and you can see very finely the diffusion of particles of ink, right? That is diffusion. Hope it's clear to you, the diffusion, right? What is the importance? Yeah, I was talking about this only. Take a glass, bowl glass, and put a one drop of ink or color, and you will see finely the particles are moving, right? Okay. Now, what are the importance of diffusion? Diffusion helps in spread of different substances throughout the cytoplasm of a cell. Osmosis is a type of diffusion where only solvent is allowed to diffuse. What is a solvent? It's a liquid form, right? Here, osmosis, whenever, like when we discuss about the osmosis in biology, it is related to the water, movement of water molecule. So, movement of water molecule from higher concentration to lower concentration is known as osmosis. Diffusion helps in exchange of respiratory gases between the cells and their environment. One more point that you have to remember, the difference between osmosis and diffusion is diffusion can take place in gas, that is in solid, liquid and gas. But osmosis only take place in solvents that are in liquid phases, right? Transpiration is diffusion process. Flowers spread aroma through diffusion to attract insects and other animals. I will give you a few more example. Diffusion, like when your mother is cooking something in the kitchen and you are sitting in the dining hall or in living room of your home, then you smell it, right? The fragrance that you smell, that is diffusion. Why the particles has moved? It is because of the diffusion. Same way, when someone spray, right, you are able to smell the fragrance, and that is also because of diffusion. The particles of those particular perfume has traveled from higher concentration from a person. The person who has applied the perfume is having more amount of concentration of the perfume particles, right? That is traveling from higher to lower. That is to us, right? That is diffusion. Now coming to osmosis. Defined as a diffusion of water or solvent across a semi-permeable membrane from a region of its higher concentration to the region of its lower concentration. Always remember, in osmosis, the difference between osmosis and diffusion, if we have to learn the difference between osmosis and the diffusion. Diffusion is simply movement of molecules from higher to lower concentration, right? Whereas in the osmosis, two words should be there in the definition, that is movement of water or the solvent across a semi-permeable membrane. So two words should be there in the definition. One is water or the solvent, other one is semi-permeable membrane, right? Plasma membrane functions as a semi-permeable membrane in case of osmosis. Here again, two more diagrams are given to you. Osmosis. You can see in the first diagram, differentiating permeable membrane is there. Sucrose solution. Sucrose solution is nothing. Sucrose is sugar, table sugar that we use at our home. So if you keep one side the water, pure water, other side the sugar solution or the salt solution, right? Instead of sucrose solution, we can also use the salt solution. We can see the movement of water from higher to lower concentration, right? This we have already discussed about the difference between osmosis and diffusion. Let's have a quick revision. It takes place in solid, liquid and gases. It does not involve any semi-permeable membrane. It equalizes the concentration of substances. Different substances diffuses depend on one another. Diffusion is not much influenced by the presence of other substances. Whereas osmosis, it takes place in liquid medium only. It requires semi-permeable membrane. It does not equalize the concentration of solvent on the two sides of the membrane. Only solvent undergo diffusion. It is influenced by the concentration and type of solutes present. Now, my students, there are different types of osmotic solution and there are different types of osmosis as well. We'll be learning one by one about them. Osmotic solutions are those solutions which can cause osmosis, which are responsible for causing osmosis. There are three types: hypotonic, isotonic, and hypertonic solution. Hypotonic solution, which has an osmotic concentration lower than another solution. Isotonic, having the similar, hypertonic having higher. Now, in the diagram, I wanted to focus on the diagram. Here, hypotonic and hypertonic. Here you can see hypo, that means water. Right, dear my student, we have to remember the amount of water. Clear? The amount of water here is more comparing to this. So the water is moving towards the lower concentration. Clear? Next is iso. Both the solutions are same, then it won't do any kind of movement. Clear? But what if one is having more, other one is having less, then water will move from higher to lower concentration. Now how to remember? Hypo means more, water is more, solute is less. Iso means solute and water concentration is similar. Hyper means osmotic concentration is higher, solute are higher comparing to water. Depending on the type of solutions, we can see two different types of osmosis. Those are end osmosis and ex osmosis. End osmosis, again, just before going through it, pause my video and remember what did I explain to you about and ex. And is when it is entering inside the cell. X is when something is removing out, moved out from the cell, withdrawn out from the cell. So similarly here, osmosis is related to water. So in and osmosis, osmotic entry of water into the cell. X-osmosis is withdrawal of water from the cell. Simple, right? When we place a cell in higher concentration, the water comes out from it. When water is coming out from it, pause my video and give the answer by your own. What is that? Right? That is removal of water. That is exosmosis. When we place the cell in lower concentration, the water enters into the cell by a process known as andosmosis. Alright. We are ending our video here. In next video, we will start with the cell wall. Thank you.